Okay, peoples. All my favorite people are here. Don't tell the people who come after you. You're my favorites. Mike West especially. Oh, but there's Ariana and Olivia and Matt and Dante.
Don't tell anybody.
All my favorites. Let's wait a couple minutes. We don't have too many people here right now. What do we got? We got 7 out of, I think, 15 or so. Let's give them a few more minutes. By the way, Celine, I was talking to, or emailing with James Faraci, and I think you're in the same category as him. He's got a minor in SLISH. He's taken all my classes. So you might want to submit that to the registrar. Dante as well. Ariana, perhaps. There she is. Nicole Lamberton. The reason I'm swaying back and forth is I'm petting the back of my huge dog right now. 115 pounds of pure love. Right, buddy? Right, buddy? What do we got? What do we got? We got 9 people. Let's wait a couple more minutes. Because honestly, I don't think we're going to take the whole hour and 15 minutes today anyway. So we'll give them a couple more minutes to come. I don't know if I told you guys this before, but I'm struggling with this online stuff because normally in class, when I'm writing on the board, that's taking up time. I'm drawing a figure, that's taking up time. Now it's like, here's the notes, here's the figure, let's move on. And my hour and 15-minute lectures are taking me about a half an hour. Bit of a struggle, I'm sure. I hope you guys don't mind anyway, that our classes are a little bit short. All the info is up there. I don't want to go too fast because then I'm not sure how well you'll be following along. Okay, I think we've got the core people here, the people who count, the people who matter. Why don't we go ahead and get started? Okay, so today we're continuing our week of Furchtgott. I'm just making a pitch for one of my favorite pharmacologists, Robert Furchtgott. My advisor in grad school worked for Dr. Furchtgott, so I have a bit of a connection to him. I'm really say two something removed from Furchtgott. What is it called? It's 2 degrees of separation from Furchtgott. Here's an interesting thing. You guys are all 1, 2, 3 steps removed from Marie Curie. Did you know that? Did you know that? Dr. Miller worked for a guy who worked for Marie Curie. Yeah, Ed Miller. Ask him. Okay, Robert Furchtgott. Today's Furchtgott lecture will be about, Furchtgott Memorial Lecture will be about nitric oxide. So him and Marty Murad and another guy won the Nobel Prize back in the 80s for discovering this, I guess you would call it a signaling molecule. Little review of what we talked about last time. Oh, I put a question up. Let's see, a quick show of hands. How many people, let's stop the share. How many people, quick show of hands, I gotta see your faces to see your hands too. How many people did the practice problem? Anybody? Anybody do the practice problem? No? Okay, so do that practice problem. We'll talk about it next week on Tuesday, okay? I did put the answer there because I want you to think about it. Okay, back to share. Sharon is Karen. Karen is Sharon. Okay, so this is what we talked about last time, the spare receptor idea. And again, this is Furchtgott. He realized that you can get 100%, 100% effect when you're comparing your dose response to your binding experiment. You can get 100% effect with only 6% of the receptors being activated, which tells them that you've got a lot of spare receptors there, receptors that You don't have to activate to get full effect. Now, why is that? Again, with your binding curve, you've got ligand plus receptor goes to ligand receptor complex. So the cell can kind of regulate how much response it gets by up and down regulating the concentration of receptor. So the body is up and down regulating the amount of ligand, and the cell is up and down regulating the amount of receptor. And between those two, they get a balance to make physiology work. Okay, here's what happens when you destroy receptors and their spare receptors. So the first curve then is just the normal dose response curve with, say, norepinephrine. And then you destroy some of the receptors by adding a certain concentration of irreversible antagonist. And that shifts the apparent potency of that, the ED50 of that agonist. You add a higher concentration, 30, and it shifts it further. And then you get to a point where you start destroying some of the receptors that are needed to get full response. And when that happens, then the maximum response starts to go down. So here, that irreversible antagonist looks like a competitive antagonist, and here it looks more like an irreversible antagonist. But the point is, you're going to get full response until you destroy all those spare receptors, and then you can't get full response anymore. Yeah. Okay, and then we talked about G protein coupled receptors and G proteins. Hopefully you guys know this, because I know you've been in most of my classes, and I talk about this in every damn class. Here is cyclic AMP. We talked about making cyclic AMP, cyclic AMP production, activation of protein kinase A, and then IP3 diacylglycerol. Again, I talk about this in every class. It's so important in cells. And here's IP3 that. Okay, and calmodulin. Calcine gets released, it activates calmodulin. So I think I do this in every class because, well, for one thing, people forget stuff from one class to another, so I have to respond. I think when I first started teaching, I would assume because you had the prerequisite of cell biology, you knew what I taught in cell biology, but people tend to forget from one semester to the next. And the other is, this is kind of like the baseline of what we're going to be talking about. We'll get into the physiology and everything, and then how the hormones affect the physiology and how drugs affect the hormones affecting the physiology. This is the bottom line, so you need to know this first before you go on to that other stuff. So I don't know, about maybe next week, maybe the week after, we'll talk about the contraction mechanism of cardiac muscle and smooth muscle, which again, I talk about in cell biology, but we'll go in more detail here, because it's all going to be important when we get into the physiology and the drugs. I like to liken it to the Karate Kid movie, you know, not the remake, which I wasn't crazy about, the original one with Ralph Macchio, how he's like waxing cars, wax on, wax on, wax on, sand off, whatever. And it's like he's getting all these, you know, kind of basic movements down and then applying them to actually being able to kill people with your hands. Okay, so nitric oxide. Nitric oxide is an extremely important regulator of arterial smooth muscle. It's also used in the brain for some signaling, but I don't know how or why, but I know it's used in the brain a little bit. It's a very reactive chemical, so it doesn't last long. I think its half-life is like 7 seconds. It took a long time to discover nitric oxide, because you couldn't isolate it. It was impossible to isolate. They used to call it EDRF, endothelium-derived relaxing factor, because, you know, before they knew what it was like, well, we know there's a factor that's relaxing smooth muscle or arterial muscle, so EDRF. And eventually they figured out it was nitric oxide. Okay, it's released from the endothelial cells. It's like produced and released in the endothelial cells, and it diffuses out of the endothelial cell into the smooth muscle cell, and it causes relaxation. So there are an awful lot of things that cause this to happen in arteries, a lot of hormones and things. I read a paper once, you know, I used to do this herbal medicine stuff, and a lot of the medicines I was using were producing nitric oxide to cause relaxation. And I read a paper where someone took, I think, about 30 or 40 different foods and tried them all, and like 90% of them produced nitric oxide in the tissue and caused it to relax. So an awful lot of things can affect nitric oxide. So how does this work? Oh man, I gotta go slower. We're almost done with today already. You've got receptors on the endothelial cells. So these receptors are things like there's muscarinic receptors there, which has always been kind of a anomaly because there are no parasympathetic neurons innervating them. They're on the inside of a vessel. There's no neurons there, but there's muscarinic receptors there for some reason, and they will cause nitric oxide release. Histamine receptors. Histamine's big in local vasodilation. So when you get stung by a bee, mast cells, which are red or white blood cells, go into the area, release histamine. Histamine produces nitric oxide, the vessels relax, and blood goes there. And that's why it looks red when something is inflamed. When you get inflammation, it looks red because you got this thing going in. Also, that histamine response is what happens if you're allergic to something, like you get stung by a bee and you're allergic to it, that histamine gets released all over your body. Nitric oxide comes out all over your body. Your blood pressure goes to 0, blood stops moving, and you die. So histamine, prostaglandin is there are several hormones called prostaglandins. They're locally made and released. They're made from arachidonic acid. So if we back up, this is your PIP2, right? Gets cleaved off to give you IP3, and now you've got these two big acyl groups here. This acyl group here, I think, or maybe it's this one, PLA2, I think it's this one. This one is arachidonic acid, which is a 20 amino acid fatty acid. I think it's got 2 double bonds. There's an enzyme called phospholipase A2, right? Phospholipase C breaks this bond, phospholipase A breaks this bond. So PLA2 gets activated sometimes and it breaks this off, and then this gets converted into prostaglandins, and they can cause relaxation. In part, here's an interesting thing for pharmacologists like you, that arachidonic acid is a substrate for an enzyme called cyclooxygenase. So cyclooxygenase will convert arachidonic acid into something else, and that eventually will become prostaglandins. That cyclooxygenase is the target for drugs like aspirin, Tylenol, ibuprofen, naproxen sodium. Like all those non-steroidal anti-inflammatory agents are going to work by blocking this enzyme that produces prostaglandins. So prostaglandins are really, they're local, but they're involved in a whole lot of different processes, and aspirin can stop their production. But here, the receptors for these prostaglandins are also on endothelial cells and can produce NO. And like I say, there's hundreds probably of things that will activate this response. But basically, you've got receptors. You know what, let's just look at the figure. You've got receptors on the endothelial cells. Excuse me. Oh, damn it. So this is the endothelial cell, and this is smooth muscle cell. You know, I meant to have a figure here to show you what the endothelial cells are. Can you guys hear this? It's the F-35s going over again. Keeping Vermont safe from New Yorkers. So your artery, okay, if this is your artery, the outside layer is connective tissue holding it together. Inside of that is smooth muscle cells to cause contraction to control the diameter. Inside of that is more connective tissue. Inside of that is your endothelial cells. So the endothelial cells are lining the vessel, so blood going through is touching those endothelial cells and nothing else. So this side of the endothelial cell then would be the lumen of the vessel, where the blood is. So we've got receptors here on the lumen side, and this whole deal here is the whole IP3 calcium thing we talked about Tuesday. So say histamine activates the histamine receptor, that then activates the G protein, GQ. GQ then activates phospholipase C, which then breaks down PIP2 into diacylglycerol and IP3. So IP3 then goes to the smooth endoplasmic reticulum, activates the IP3 receptor, and calcium comes out. So all of that is what we talked about already. But here's where it changes. So when the calcium comes out in these endothelial cells and binds to calmodulin, Calmodulin is going to activate this enzyme called nitric oxide synthase. And with a name like nitric oxide synthase, you can kind of guess what it's going to do. It's going to synthesize nitric oxide. So it's going to take arginine, the pirate's favorite amino acid. If you know the structure of arginine, it's like, you've got your backbone like every amino acid, and then there's a bunch of carbons come out, and there's like a double bond nitrogen and a single bond nitrogen or something like that out there. So there's a lot of nitrogen on the side chain of arginine. So NOS is going to take the nitrogen off arginine and make nitric oxide. Byproduct citrulline, that's the other part of the molecule that's left, but we don't really care about that because it doesn't do anything as far as I know. So anyway, nitric oxide synthase is going to take arginine and produce nitric oxide. Nitric oxide, small molecule, can diffuse anywhere it wants. It diffuses diffuses from the endothelial cells to the smooth muscle cell, and there it encounters guanyl cyclase. The guanyl cyclase is like adenyl cyclase, except it's going to convert GTP into cyclic GMP. So again, very similar to the cyclic AMP mechanism, except we're doing it with GTP. So guanyl cyclase makes cyclic GMP, and cyclic GMP will then activate protein kinase G, which is similar again to adenyl cyclase. I think one difference here is, if you remember adenyl cyclase, it had 2 regulatory and 2 catalytic subunits. And when cyclic A bound, the 2 regulatory came off together and the 2 catalytic separated and they went off and did their thing. Here with cyclic G, you've got 2 regulatory and 2 catalytic, but they separate, and the catalytic is like a homodimer, I believe. I could be wrong. It's not important. But anyway, it's still a little different, but the same. The main thing that's different about protein kinase G is it's got different consensus sites. I don't have it here. I have it in my notes. It's a little bit different than cyclic A, so it's going to phosphorylate different targets than cyclic A. Some of the same targets and some different targets. So we'll talk later about the effect of beta-adrenergic agonists on smooth muscle cause relaxation by phosphorylating specific proteins. Cyclic G will phosphorylate, G kinase will phosphorylate the same targets, but also several other ones. So NO can really relax smooth muscle more than a beta agonist can. Does that make sense? Yes? No? Yes? Yes, okay. All right, so that's our nitric oxide. You've got receptors on the endothelial cells, they get activated, and that turns on the whole IP3 calcium thing. Calcium gets released from the smooth ER and binds to calmodulin. Calcium calmodulin activates nitric oxide synthase. NOS then uses arginine to make nitric oxide. NO diffuses into the smooth muscle, activates guanyl cyclase, makes cyclic G. Cyclic G activates protein kinase G. And in terms of turning this mechanism off, nitric oxide has got a very short half-life. I think it reacts with like everything, like water, and everything in the cell is going to react with. So you don't have to worry about that going away. It will. So that's the main thing in turning this off. And also you've got phosphodiesterases that will. Oh, there's another interesting drug. Phosphodiesterases will block this cyclic GMP or break down cyclic GMP, and that will shut off PKG. So here's another interesting pharmacological connection. I talk about this in every class because it's kind of serendipitous how these drugs get found sometimes. So people were working on drugs to block guanyl, block the phosphodiesterase that breaks down cyclic GMP, because they're thinking if we can block that, then cyclic G will increase. It will cause arterial relaxation and decreased blood pressure. So they're looking for blood pressure mechanisms, blood pressure drugs that will cause relaxation of smooth muscle. So they came up with this one, sildenafil or something like that, and they tried it in some healthy volunteers, which is your first trial, and they're like, well, didn't do much to my blood pressure, but let me tell you about this other side effect. It was it turns out cyclic G is really important in the arteries leading to the genitals. So when they increase cyclic G, more blood flow to the genitals, and if you're a male, up goes Mr. Winky. So that became the drug for erectile dysfunction of Viagra. You know it as Viagra, but it was found accidentally when people were looking for blood pressure medications. Kind of interesting. There's another similar thing where there was one drug that I think, what did it do? Block potassium channels, I think, and they thought it might be good for blood pressure or activate calcium or potassium channels. And it turned out it didn't really have much effect on blood pressure, but it makes you really hairy. So that's minoxidil. You put that on your head if you've got enough money to afford the monthly payment. You put that on your head and it makes your hair grow back, minoxidil. Another kind of blood pressure mechanism that serendipitously did something else. So I'm just filling time here. I hope you're not offended by this. Everybody's offended anymore. I'm offended. Okay. Okay, everybody good with NO? Everybody happy? Yes? Okay. Not that complicated, but it's kind of adding a little extra something to our day. Okay, synapses. Again, most of you, I think, were in my drugs class. We talk about synapses a lot, but I need to go over it because this is where most of our drugs are going to work, in the synapses. So the synapse then is a gap between the neuron and the effector. The effector is like the end organ. So in terms of effector in this class, we're talking about, smooth muscle or the heart or whatever it is that the motor neurons are interacting with. That's the effector. And another plug for a pharmacologist, Otto Lowy, I think he was a German Jew in the 30s, came across or did the definitive experiments that showed that nerves release a chemical that causes the activation of the effector organ. So he, in the 1930s, he proved that, you know, the transmission of information from the nerve to the muscle was through a chemical and not a direct electrical stimulation. He did this by taking a heart with the vagus nerve still attached to it, put it in a chamber so there's, you know, it's surrounded by water, stimulating that vagus nerve, and the heart rate slowed down. And then he took some of that fluid out of the chamber, put it into a different heart, and that heart slowed down too, showing that there was something chemical transferred from one to the other. And then towards the end of the 1930s in Germany, this poor little Jewish guy got the hell out. You know, it just doesn't make any sense when you think about it. Like there were so many just talented Jewish scientists and musicians and everything. And World War II. Anyway. All right, effectors. What are we talking about? Arteries, veins, the heart, and the ever popular, etc. You probably know this by now. I put etc. when I don't feel like talking about it anymore. It's just, yeah, over there. Yada, yada, yada. The exception, when am I saying this? Oh, the exception with the effector rule is autonomic ganglia, which we'll talk about. So in the ganglia, it's like the neuron from your medulla comes down your spine, goes out the ventral root of the spine and interacts with another neuron. And where it's turning another neuron on, that's the ganglia. And then that goes off, and that's the motor neuron goes off and activates the effector over there. Okay, so mostly what we're talking about is the neuroeffector junction, but we'll talk about the ganglia as well, which is the only nerve-to-nerve connection we talk about in this class. Okay, connections between the CNS and the motor neurons. Okay, so that's your ganglia. Okay, again, if you've taken my drugs class, you'll recognize this because this is the exact same figure we talked about there. So what's going on here? Over here, these yellow bars are microtubules. So the microtubules are coming all the way from the cell body, you know, just near the Golgi apparatus. So these vesicles with enzymes and things in them are being made in the cell body. That's where proteins are made. So they get made there, packaged into vesicles, put a motor map on it, and that drags it down the microtubules all the way to the axon bulb. This is your axon bulb here. Imagine this: you've got a neuron, motor neuron in the base of your spine that sends an axon from there all the way to the end of your toe. Okay, so these axons are really, really long, and these vesicles come from way the hell back there all the way up. So anyway, these vesicles then have got a number of things in them. They've got an amine pump and some enzymes inside them. So the amine pump, or not, I guess, let's just call it a pump because it may not be only amines. So the pump is putting either recycled neurotransmitter back into the vesicle or precursors to the neurotransmitter in the vesicle. And those precursors then will be converted into neurotransmitter by the enzyme. Oh, shit. Damn it to hell. The precursors will be converted into neurotransmitter inside the vesicle. So basically these vesicles are going to be packed full of neurotransmitter. And they're going to be attached to cytoskeleton. And you've got them kind of in ranks here, in rows. And the very last row will kind of release from the cytoskeleton and attach to other proteins right on the presynaptic membrane there. So there's a number of proteins that are going to hold it in place, and then it primes and gets ready to fuse with the membrane. And they just sit there waiting for an action potential to come. So when the action potential comes down the axon, it reaches the presynaptic membrane, and that depolarization causes the voltage-dependent calcium channels here to open. There are, I think, 4 different types of voltage-dependent calcium channels. These are the N type. They'll be different from the ones we see in the heart. So our heart drugs don't affect this, neither here nor there. So this voltage-dependent calcium channel then will open, calcium comes in, and that causes these vesicles to fuse with the membrane and release that neurotransmitter into the synapse. So then the synapse gets filled with neurotransmitter, and it will diffuse to the other side and activate receptors on the opposite side. So that's what we talked about last time, activating beta receptors, if it's epinephrine or norepinephrine, to cause relaxation, or activating alpha receptors to cause contraction, or activating muscarinic receptors if it's a parasympathetic neuron, causing whatever it is that does. So that's what we talked about already, what's going on over here. Okay, so every car has got both a gas and a brake, right? You can't just have a gas pedal. So how do you turn this off? This signal then is turned off by the amine pump here in the membrane. It's actually spread all over the membrane, but I put it over here. And that amine pump then it's a indirect active transport. It uses the sodium gradient to pull the amine in with it. So it's a co-transport mechanism. So it brings the amine in, and then now the neurotransmitter in, and now you've got neurotransmitter all, through this cytoplasm here of the axon bulb, and then these pumps then put it back into the vesicle, recycling the neurotransmitter like I was talking about before. And if you think about it, if these things keep fusing, if these vesicles keep fusing, that axon bulb membrane is going to get bigger and bigger and bigger and bigger. Pretty soon it's going to run out of vesicles too. So you need to recycle the membrane as well. So you've got this endocytosis process going on as well, pulling membrane back in, pulling these amine pumps back in, because when that vesicle fuses, that goes into the membrane as well. All those enzymes that are inside the vesicle are membrane attached. They become part of the membrane of the axon bulb. So all that has to be like endocytosed to bring it back in. So we're recycling vesicles at the same time we're recycling the neurotransmitter separately, but at the same time. And then these vesicles get put back into order, and then they can be used again. So everything gets recycled. One more thing to talk about is our autoreceptor here. So the autoreceptor is going to respond to the neurotransmitter as well. So you've got your presynaptic autoreceptors, and that will then inhibit the voltage-dependent calcium channel. I'm pretty sure mostly the way it's doing that is by blocking the production of cyclic AMP. Cyclic AMP is important for regulating these calcium channels. So, but don't worry about that right now. Basically, your autoreceptor is blocking, or not totally blocking, but modulating these voltage-dependent calcium channels. So when you've got neurotransmitter out here, you get less calcium coming in through the VDCC for every action potential. So you get less neurotransmitter being released. So it's a negative feedback mechanism, blocking blocking too much release of neurotransmitter. Oh my God, I'm done already. Holy snapping our souls. Okay. Just looking over my notes here, see if I forgot anything. Oh, one more thing, which you probably know already, but I should say it anyway, is that mechanism is mainly for us anyway, norepinephrine, because we're mainly going to be talking about norepinephrine and acetylcholine. So that's norepinephrine. Acetylcholine has got an enzyme here in the synapse called acetylcholinesterase, and acetylcholinesterase will acetylcholinesterase will break down into acetylcholine into acetate and choline, and those will both be recycled back into the membrane through pumps like this, and then recycled back into the vesicle and made back into acetylcholine. So, Mike, I'm getting seasick. Mike's moving around.
Sorry about that. Dogs are barking.
Hey, I'm just teasing you. I need to kill time because we're only half an hour into this class. You know what? Why don't we do this? Okay, let's just stop our share. Any questions about what we talked about today? That's pretty much what I want to talk about. Like normally in class, that would take me the whole hour and 15 minutes. Are we good with this? All right, well then let's go to that problem I put up in Moodle, and talk about it. Oops, I'm just bringing it up and then I'll share it with you. Oh, here it is. Oh, 6 people did attempt it. Good. Share that. Sharing. Okay. No. Is this the question? Oh, I guess so. How does a partial agonist compare to a full agonist in a tissue with a large amount of spare receptors, more than 95% spare receptors? Draw a dose response curve with both of them on it, assuming they have the same KD. So let's see if we have any brave people here. Just copy this so I don't lose it. Okay, question is, how does a partial agonist compare to a full agonist in a tissue with a large amount of spare receptors? So say you've got 2 agonists, one is partial and one is full, and you're doing dose response curves with them in a tissue that's got a lot of spare receptors. Their KD is exactly the same. What's going to be the difference in the dose response between the partial and the full, do you think? I'm going to have to bother people. No one's going to answer. Ariana, let's see your faces. Matt Gremlin, why don't you bring your face up here? Oh, there's Ariana. Olivia. Matt, we don't want to see you. Frankly, you can leave it off. Ariana, what do you think? So you've got 2 agonists. They have the same KD, okay? Partial agonist and full agonist. You're going to do a dose response, spare receptors. A lot of moving parts here. What do you think?
Maybe the partial agonist would take a while to get to 100% response after the spare receptors are added. So it would have a lower, maybe like ED50.
Bingo. Exactly. So partial agonists, you usually say you don't get full effect, but with spare receptors, you will get full effect. But because you need to occupy more receptors to get that full effect, the ED50 is going to be more rightward shifted. Exactly. Okay. And then I I guess if you started destroying receptors, you'd find it, well, it would end up looking the same. Never mind. That's not that important. Okay. Good answer.
Could you repeat the answer again, please?
Sure.
Thank you.
Let me pull up a figure, and it's not exactly right, but it will help me to explain this. Okay. Okay, so it'll look kind of like this, not all of this. Okay, so ignore all this IA stuff. Okay, get rid of that. And just look at these two curves. So both these drugs are binding at the same rate. Okay, the binding of the drug to the receptor is the same. The full agonist, though, can get you to full effect at a lower concentration because every receptor that it binds to is going to give you 100%. So you only have to bind 5% of the receptors in the tissue to get 100% response. Here's your partial agonist, or probably this one over here, where normally you would say a partial agonist would be like this. It can't really get to 100%. But because there's spare receptors, let's say your partial agonist normally, if there's no spare receptors, can get you to 30%, right? That means theoretically each receptor is being activated to the level of 30%. Well, here with spare receptors, you can activate more receptors to 30% and still get up to 100% effect because you've got so many receptors there that the partial agonist can still get you to 100%. But even though their binding kinetics are the same, it's going to take more partial agonists to get you to 100% because you have to activate more receptors to get there because you're not fully activating each receptors. Beeswit, does that make sense, or you want me to say it again? Good? Okay. Oops, stop the share. Okay. Okay, what time is it? 10:06. Let me make a quick plug here. I'm not wearing a t-shirt just to be informal. I'm gonna make a quick plug for SUNY Plattsburgh Pre-Med Association. Yay. James Faraci, I think, is still president. He'll probably be seeing this later. He's in the class, but Olivia's there. Yay, Olivia.
He is MVP.
What's that? What did you say?
I said he's still president and MVP.
Oh, good. So Olivia's VP.
So 7 p.m. on Tuesdays.
Hey, 7 p.m. on Tuesdays. So if you're interested in a health career, hook up with Olivia or James and go and I guess virtually go to the meetings. I don't know. How are you doing meetings this year?
They're going to be bi-weekly on Zoom, and the meeting ID numbers are on, James sends them out in an email, and they're also going to be posted on the Instagram page at PSU Pre-Med Pre-Health.
Okay. Could you send me that information in an email? Because I'm old. I only do things by email.
Sounds good. I'll put the tag in the chat section.
Okay, and I'll try and publicize it more to get more people out there. Okay, I think that's all I got for today. I hate to say this, but I'm done already. Are you guys good? Yes? We good? This is yes. Okay. So next week we'll be doing more of this, I believe, and then I guess I'm going to have to. I've got that paper I wanted to do, but the school in a budget, in a money-saving move, it made it impossible for me to get it by PDF. I'll see if I can still get it by PDF and send it to you because I'd really like to do a little bit of that reading primary literature so you can get experience with that. That's always something that is fun. Don't those curves have different KD values, or am I reading the graph wrong? Oh, so Matt, no, because they don't have different KD values. We've got different ED50s because those are dose response. They're not binding. Okay, and that's kind of the point. The KD of both drugs was the same, but the ED50s are going to be different because the partial, you have to activate more receptors to get it. Can someone please tell him to peep the chat? Oh, thank you, Matt. I just peeped it. Okay. So Olivia, what do I do with at PSU Pre-Med Health? Do I go to Instagram or something? You're muted. You're muted. Unmute yourself there. Oh shit, I just muted you.
That's the Instagram handle name. So if you go on Instagram, that's the username that you'll.
Okay. Okay. So, but if I set up an Instagram account, is that like another hour out of my day that I'm going to be looking at Instagram? I mean, Facebook's already taken up 4 hours of my day. I don't know if I can handle more.
Now if you follow us, we only post it probably once every couple days.
I'll only follow you. Okay. All right. So I guess we're done for today. Thank you for coming. I hope to see you again next Tuesday at 9:30. Bye, guys.